So thank you so much, Jim. I'm really honored to be the recipient of this award. Usually my students say I break everything. Here the students broke my presentation, just for the record. So thank you. I'm really honored to be receiving this award and really honored to be receiving it or being given the award together with Professor Deckard, who's made some fantastic contributions as well. I realize most of you are here to hear him speak, but bear with me for 30 minutes and we'll try to tell you something about IR imaging and Raman imaging as well. So I'm going to tell you something about a few directions in my lab that are ongoing right now. And they relate on two ends. One to something very fundamental. It's to the theory of IR microscopy, and something very, very applied, how we determine therapy in cases of advanced cancer, for example. So I'll start off from fundamentals, and my lab is also engaged in looking at Raman spectroscopy and theory and modeling in that arena as well. But I won't talk about that at all in today's talk. Instead, we'll focus on the infrared part of life. And what I'll do is focus a little bit on the infrared imaging aspect. So as you've all heard perhaps in this meeting, that we can now get pretty nice data sets that look like this. This is essentially an X and Y image, and then in the Z domain, there's a spectrum. So getting spectra out or getting images out now is fairly routine. In my opinion, the challenge is, how do we take this data set and turn it into knowledge? How do we take it and turn it into knowledge that's actionable, actually, which is perhaps a greater challenge. So what we've been doing is we've been involved in trying to understand more and more how this data set is generated, what are the fundamental underpinnings of the data that we actually record, and then use that to perhaps better utilize the data for something useful. So I'll take you back to a few years ago. This is some data. This is some data that we worked up almost 5 or 6 years ago, and we didn't understand this at all. So what we did here was we took a population of patients, and we took multiple samples from the same patients. All of these patients were from different clinical settings. There were a number of samples from them, and then we performed FTIR imaging on them. So now we have a data set for each sample from each patient from each clinical setting. And this pretty much involved all the variability that we could ever think about in IR imaging data. And then we ran a very simple ANOVA model on this and said, if we look at the spectral features, so this is a combination of features that one can look at in the entire spectrum, we found that some of them were discriminative for disease, or in this particular case, for cell type in the tissue. So that's the good news. So there's a subset of the infrared spectrum which allows us to discriminate between different kinds of cells that are physiologically important in the body. And then we saw contributions that were distinct for different spectral peaks arising from different factors, whether they came from different clinics, whether they came from different patients, whether they came from the same patient but different samples. And there was, at the end of all of this, there was one factor that we couldn't explain. It's these gray dots there, or gray inverted triangles. And this factor that we couldn't explain, this residual, which should ordinarily be very, very small, actually started to dominate after a while. It became so prominent that we couldn't even use the data for doing any kind of physiologic monitoring. So the question was, where is this coming from, and what was happening here? And this had us confused for a while. So if you keep that in mind for a second and start looking at where the data actually comes from in the infrared, that's perhaps more interesting. I think more realistically, when I started as an assistant professor, I was convinced that I would never be funded. So I started doing theory quite a bit. And I started going philosophical. I started asking questions like, what is the basis of IR imaging, and what kind of data that we acquire, and so on. So this was a piece of evidence that we were aware of, but never sort of could solve it. At the same time, we were thinking about what are the fundamentals of IR microscopy. And I'll give you a very, very simple equation. The fundamentals of IR spectroscopy in general are based on this. It says that the absorbance that you record is measured by the intensity that is transmitted with and without the sample. It's fairly trivial. This is Beer's law. It's been around for a long, long, long time. But what they don't tell you is that there's this little factor. And this little factor comes from whether the transmittance function of the sample, and in fact, indeed, the whole system remains constant or not between the two measurements. If it doesn't remain constant, then this term obviously is not zero. That doesn't cancel out. And you actually start to get a contribution from this term that becomes more and more significant. So what you see here, the whole game about understanding data that you record and how it deviates from sort of the standard is based on essentially this. So if you think about it, what Beer's law tells you is that you have a cuvette of liquid, or you have something that is entirely non-scattering. Your light comes in at a right angle and goes through that material without scattering, without going out of your beam path. So that's what Beer's law says. If you look at the optical microscopy literature, what they tell you is light comes in, gets scattered once, sort of the Born approximation, and then you collect that light and you deal with it. But no one tells you what happens when absorption and scattering happen at the same time. And that's really the crux of this issue. So what we've done is over the last 4 or 5 years, there's a series of papers in analytical chemistry. We've published everything in one sort of journal, so you can go back and look at it. What we've done is systematically gone through the process and modeled the IR microscope from first principles, from the source to the detector. So now in the lab, we have something, we have a model, we have a framework in which everything that you can ever imagine in an IR microscope is modeled analytically. So this whole thing exists, and this framework provides some very interesting results. So I won't belabor the 4 or 5 years of effort there. You can read about it, but I'll share with you 2 rather important results from this. The first one is something that is of fairly contemporary interest. So what happens when you take your microscope and put a sphere in it? Okay, it's a simple thing. You take a polystyrene sphere. These are fairly consistent sizes. Everyone knows what they look like. And you put it in a sphere, and the data you record actually look nothing like polystyrene. So the electric field and the light that is the intensity that is transmitted look like this, which is perhaps less interesting to this audience. But what is more interesting is this is exactly the same sphere. This is PMMA. And all I'm doing here is changing the radius of the sphere. So when I change the radius of the sphere and compare it to sort of the ideal case, the same volume of the sphere, which would be recorded in say, a film that was normal to the beam, it looks something like this. So black is the spectrum that you expect, and red and blue are the spectra that you actually acquire because of size effects. So what we've been able to do is explain this, right? So we have a forward model that actually predicts what the data will be if you were to place a sphere in it, and that agrees quite well with the recorded data. But then we went one step further. We said, what if you have data that you were recorded and you knew nothing about the material? Then what would you do? Well, it turns out you can use the same framework and actually invert this problem. This is quite well posed in this particular case. And when you invert it, you can not only get the size of the sphere, but you can also get the real and imaginary part of the refractive indices, right? And once you have that, once you have size, once you have imaginary and real parts, then of course you can reconstruct whatever you like from this. So this is perhaps the most challenging problem in IR microscopy that we could think of. It seems rather trivial, but this is actually a sphere that is of the same size as the wavelength of light that you're using, and it's actually a three-dimensional object. So it actually turns out to be a rather complicated thing. But what saves us here is the fact that we know it's a sphere, so we can really predict things quite easily and so on. We also have a full framework now to predict things if it wasn't a sphere, if it was some arbitrary object, if it were a piece of tissue, for example, and we can do that. So the same ideas hold, but that's an even longer story, so I won't go there. The next aspect that I'll discuss with you is trying to extract more and more information. So here what you saw was we were trying to extract more and more spectral information. We weren't necessarily so concerned about what it looked like spatially. You can do the same thing. This is imaging. So you can try and extract more and more spatial information rather than spectral information. And that's been a second direction that we've sort of pursued. So this is, again, slightly older data, and this is an old story now. So on the left-hand side in all of these images, you'll see sort of state-of-the-art commercial instrumentation, which gives you this sort of imaging quality. So this is using a spotlight spectrometer. This is using a variant spectrometer. And this, I believe, is using a Bruker spectrometer. So just to let you know, we're not biased. And this is exactly what you get with every single piece of instrumentation that's out there. So with Carol Hirschmugel, a couple of years ago, we published this. And what we did here was reduce the pixel size down to a level that was thought unimaginable. So this is about 5.5 microns per pixel, and this is half a micron per pixel. And you will see right away that this is not a fool's errand. We actually get better quality data from this. And the same thing happens. This was implemented on a synchrotron. This is implemented in our lab, and this is also implemented in our lab. So now this, what we call high definition imaging at that point, is now fairly routine. And there's actually a paper that came out earlier this year which tells you why and how you're able to get this sort of imaging. But I just want to reiterate that point. That this is due to a number of factors. It's due to using a higher NA lens. So this is the recorded data with sort of a lower NA lens. So we used a higher NA, this is the higher NA lens and this is the lower NA lens. So we used a higher NA lens, number one. But the second thing that we did that was interesting here was we used the appropriate pixel size for the data that we were recording. And it turns out the considerations of spectral localization for chemical imaging, or where there are spectra associated, compared to optical imaging or microscopy are actually different. The resolution criteria are different. The information you can extract from a certain pixel size is different, and you can read all about it in this paper. But what we were down to here was sort of a micron scale imaging. So if you're interested in let me just summarize, if you're interested in imaging wide field images in the far field in IR microscopy, you should be using pixel size that are about 1/10 of the wavelength roughly, and you'll still get enough information out of that data, but you can read about it on here. So this is still older stuff. There's some even newer thought. So let me ask another question. What if we were not interested in wide field imaging, but if we knew that in a given focal volume we had multiple particles that were different in spectra? Then how far could we push this, right? So the spectral axis afford us a discrimination that we didn't think about before. And so we started asking that question. And really the key here is how much spectral information can we use to extract spatial information, and how much spatial information can be improved if we have spectral information. So this interplay is not usually available in other forms of microscopy, but certainly in this case it is. And I'll give you a very brief example. So this is a piece of theoretical work. We haven't implemented this quite as nicely yet. But here what you have are three species in the same focal volume. So you have think about it, three small spheres, for example, 3 nanospheres sitting in the same focal volume of a far field microscope. And what we're doing is illuminating them for a given, so there's a spot size here and the 3 are sitting there. And the question is that if we know that there are an arbitrary number of particles sitting in a focal volume, can we recover the centroids of each of those particles? Okay, so we have to be careful here. We're not claiming I can recover the shape of those particles. I'm just saying, can we localize the centroid of chemically distinct species in a focal volume using spectral data? And here it turns out that if we think of these as spheres, for example, we've modeled these as spheres, it turns out you have lots of information, you have the so-called spectral distortion, which is really nothing but the effect of morphology on these spheres. So even if you're not thinking of chemistry, even if you're thinking of simple things like morphology and you have multiple spheres, what you can do is use their scattering functions from the same spatial volume and actually localize their positions. So here what you see are 4 spheres that we've arranged in different sorts of configurations. And based on their spectra, we can tell you where the location of the centroid is and what is the spectrum associated with each point scatterer in this particular case. So if you think about this framework, what it tells you is that if you have n species in a given focal volume, you can localize with almost infinite precision those n species if they're orthogonal, spectroscopically orthogonal. If they're not orthogonal, then the trading rules become something that is a little bit complicated, that depends on the spectral overlap, and that depends on the signal-to-noise ratio. So the trading rules for IR microscopy, I think, are really, really different compared to optical microscopy, and it's something that is still an open frontier, and we don't know how far we can push this. I think the best analog to think about this sort of approach is to think about single molecule or in fluorescence, what is sparsely illuminated and recorded in sort of super resolution mapping, the palm storm type of approaches. So you can do that not just in time domain, here we are showing you can also do that in spectral domain. Of course, you could do this in Raman imaging, which is also something we're trying, and all you would have to have here are nanoparticles, for example, and then you can localize within a given focal volume where each particle is, provided there are some spectral differences in those particles. So with this, I think I'll switch gears here a little bit. Do I have 5 minutes or so, Jim? Is somebody keeping time? Because I'm not, sorry. So I'll switch gears here a little bit and tell you about where we're applying these strategies that we've been developing. So what we're interested in my lab is trying to look at cancerous samples and trying to answer questions at different levels in the healthcare enterprise in the cancer domain. So if you think about the cancer diagnosis and staging problem in this country, we take a large number of asymptomatic people and put them through what we call screening. And then the only question there is to say, is there a suspicious lump there or not? Is there something that we need to do afterwards? If there is, then we go in for a biopsy. So we take millions of people here for breast and prostate cancer each. We roughly have about 1.2 million people for biopsy. And then we ask a very simple question. We say, is there cancer or not? And out of which the answer for about 230,000 people is unfortunately yes. So we have to diagnose their cancer. We have to say, what is the grade, what is the stage? And then for some of them, we say, is more therapy needed, and so on. And of course, there's an increasing number of survivors, so we have to keep monitoring them over time. And so what my lab has done is trying to focus on each of these problems and try to build solutions that are based essentially around looking at microstructure and sort of microscopic compositional changes. So if you look at the clinical gold standard from this stage to this stage, it pretty much relies on taking a piece of tissue from the human body, slicing it thin, putting it on a glass slide, and putting some dyes in it. And then a pathologist goes in and circles and says, here's a cancer, and this is normal tissue. So again, in a nutshell, what we try to do in our lab is to use IR imaging and image this unstained tissue. So go one step before this, image the tissue before it is stained, and use computation, use chemometric algorithms in the background to try and say what each pixel is in terms of its cell type and in terms of the disease that might be present in every pixel. So again, this is a larger story. But the idea here is that can chemistry and structure add up in a way that is better than structure alone? So again, to place this problem in perspective, there are a number of issues in this problem. So here's a chart of what happens in prostate cancer in this country. If you have 1,000 men who are screened, then about 100 to 120 of them have a false positive result. So they're unnecessarily told that perhaps there's a suspicion of cancer there. 110 of them roughly get diagnosed with cancer. We treat 97% of these people quite aggressively with surgery, with drugs that are quite harmful. And at least 50 have side effects from those drugs that are long-term, that are problematic on a daily basis, that significantly degrade the quality of life. 4 or 5 people out of these 110 still die from prostate cancer, and it is estimated that between 0 and 1 life is saved for every 1,000 people who are screened. Okay? So the real problem in prostate cancer is not the fact that you're actually getting prostate cancer, or perhaps you might die from it, which is only significant for the 4 or 5 people there. But it's really, what do we do about people at all these stages? Can we predict who this lucky person is and really treat them? Because for these people, even our best treatments don't have hope right now. Can we not treat these 50 people because it's going to be quite dangerous to treat them? And can we better identify these 110 people from these 1,000 people? So there are many, many questions at different stages in the prostate sort of enterprise. And we've tried to attack all of these by sort of putting them in a common framework of tissue analysis. So what we've done is given sort of 4 levels of analysis that we work at in my lab. The first one is just looking at efficiently large numbers of tissue that are screened in the clinic. The second one is to prevent errors. So can we make sure that every 110 of these truly belongs here, and even every 100 or 120 of these is correctly classified? The third problem is a little bit more complicated because determining therapy and determining which kind of therapy here requires multiple diagnoses from multiple settings. So can we take an expert pathologist in a box sort of approach and put it into a computer and have that available in every hospital? And the last thing is something that is just absolutely blue sky. Can we far exceed the prognostic capability of current clinical methods and do that well enough? So this is, again, a pretty long story, but I'll cut to the chase here and give you some very recent data that looks like this. Let me forward out. But this is something from my students' poster that was there. But let me highlight this one study that's recently under review. So what we did here was we took 250 prostate cancer patients who all died within 5 years, unfortunately, of the disease. And then we took every clinical predictor of those patients and found a matching twin who survived more than 10 years, right? So we matched up these 2 data sets, and there's absolutely no way to tell this apart using any genomic, proteomic, or any other approach in the clinic today. And we actually took those 2 data sets and said, can you place them in group 1, which is the dangerous group, and group 2, which is sort of the non-dangerous group who would not die from the disease? And what we have here is the current clinical method. So Capra-S and Catana are the 2 currently used clinical gold standards. And if you look at them, the area under the curve, their sensitivity and specificity is exactly 50% roughly each in these 2 cases. It's like tossing a coin with the best methods. And what we've been able to do here is use IR imaging data and actually exceed this prognostic capability of both these methods. Not only that, we could also show here that the IR imaging data is actually independent of the Catana and Capra scores that are being used right now in the clinic. So if we add the Catana and Capra back, so only in cases where Catana and Capra fail does IR imaging provide a benefit in this particular case. So if we add this in to Catana and Capra, perhaps the joint test could improve prostate cancer diagnosis even more. But what is more important is we can give two pieces of information that are not available in the clinic right now. The first thing we can do is take this integrated IR imaging approach and provide an odds ratio. So if you're in the low risk group, let's say your odds of getting prostate cancer are something, but if you're in the high risk group, we have a 20-fold higher chance of having lethal prostate cancer if you're classified by IR microscopy. The best clinical tools give you a 6 and a 2-fold roughly chance of getting between the high and the low group. So in CAPRA, what is really useful in CAPRA is discriminating this group from this group, right? So the ratio between this to this is 6-fold, but again, even moderate to this is only 3-fold. So it's roughly a 2-fold advantage between low grade, moderate grade, and high grade. Catan is worse, which is the sort of gold standard on the East Coast, which is just a 2-fold increase between these two. We can certainly improve these results 10-fold, as you've seen here. We can also point out where in the tissue we're getting this discrimination from. So what we're doing in our lab now is going and cutting out these places that IR imaging has identified as being dangerous in the tissue and looking at the genomic analysis and other molecular techniques with this. So if we can see, if we can figure out where things are going wrong and where it's coming through. So with that, I think I'd like to stop here, except for if I can get this to move. Just lead to one more thing and say that really it's this combination of instrumentation, theory and algorithms, and in some ways partnering with people who are interested in real problems that can lead to benefits. And in this particular case, I hope to tell you that activity in this area is leading directly to patient benefit, and we hope that will be used sometime soon. So thank you again, and I'm really honored to be here. Thanks so much.